Non-fermenting gram-negative bacilli. So the patient is a 25-year-old female. Patient history: received a bone marrow transplant as a treatment for aplastic anemia. Fever, chills, and malaise for about 2 days duration. Initial laboratory specimen obtained. Blood cultures, 2 sets, 20 mils of blood in each. Second set of blood cultures were collected 5 hours later. Blood culture bottles were positive for gram-negative bacillus, oxidase positive, non-lactose fermenter. Identified the gram-negative bacilli using mass spectrophotometric methods. Antibiotic susceptibility tests performed thereafter. So definitions: fermentative or fermenters are organisms that can ferment carbohydrates. Non-fermentative or non-fermenters are organisms that do not ferment carbohydrates. General characteristics: most group members are oxidase positive. Some oxidize carbohydrates to derive energy for their metabolism, and they're referred to as oxidizers. Some group members do not break down carbohydrates at all. They're referred to as non-oxidizers or asaccharolytic. Anytime you put the A in front of something, it's like the absence of, or it's missing. Saccharo is talking about like saccharides, and saccharides are sugars. So asaccharolytic, and lytic means to break apart or cut up. So they have the inability to break down saccharides. Fail to acidify oxidative fermentative OF media when overlaid with mineral oil. And they failed to acidify triple sugar iron TSI agar butts. They're found in most environments, typically soil and water, on plants and decaying vegetation, and in many foodstuffs. They prefer moist environments. They prefer and grow much better in aerobic environment. Non-fermenting, long, thin gram-negative bacilli. They tend to be resistant to multiple classes of antimicrobials. They account for about 15% of all gram-negative bacilli isolated from clinical specimens. They're responsible for a number of serious infections, usually following surgery or trauma, septicemia, meningitis, osteomyelitis, or wound infections. Risk factors for disease. So risk factors include immunosuppression, trauma, foreign body implantation, or infused fluids. Characteristic clues that can indicate the presence of a non-fermenter in a clinical isolate: oxidase positive, may be weak and variable. Non-reactivity in 24 hours in commercial multi-test kit systems. No acid production in the slant or butt of TSI agar or Klingler iron agar, KIA, and resistance to a variety of classes of antimicrobials. Classification systems: there are many. One system uses the reactions of 3 common tests: growth on McConkey agar, oxidase reaction, and glucose OF tests. The glucose OF test looks like this. So typically you would inoculate 2 tubes. One tube you would kind of keep open, and this is known as the oxidative tube. And when it's open, you kind of loosen the cap on the top so that oxygen can get in. The next tube you're going to overlay with mineral oil, and then you seal it. So when you look at the results, yellow means that it's able to utilize the sugar in that condition. So in this first one, it says A is fermentative and oxidative, so it can utilize the sugar under fermentative conditions in the absence of oxygen, and under oxidative conditions in the presence of oxygen. For B, it's just oxidative, so it can't ferment, but it can utilize the sugar in oxidative conditions. In C, glucose is not metabolized. It's called inert, also asaccharolytic. So here they're both green, which means the glucose was not utilized. When glucose is utilized, an acid is produced, which changes the pH of the media, and the pH indicator will then change color. So when you see the yellow color change, you know it's positive for acid production, or for the utilization of that sugar. When it doesn't change color, it's just going to be negative. This is a really good flowchart that kind of shows the differential results of various species here. You do not need to know all of these different species, but a lot of these are ones that we've talked about before. So kind of pause the video and take a look at these and see some of the differences here. But here it shows the McConkey agar results, the oxidase results, and then whether it's an oxidizer or non-oxidizer. Genera that make up the most isolates seen in clinical laboratories would be Pseudomonas aeruginosa, Acinetobacter species, Burkholderia species, and Centrophomonas multifila. These 4 species are the basis for one of the lab activities that we do in class. Some general characteristics are listed here, so you can take a look at these and review them. Guidelines for identifying non-fermenters. So the decision to identify many of the non-fermenters is based on the site from which they've been isolated. That is, whether they have been recovered from a normally sterile site in pure culture or from a non-sterile site in which 3 or 4 other bacterial species are also present. So if they are isolated from a sterile site, then we definitely want to work them up through the identification. Identification methods, MALDI is typically the most common. Susceptibility testing is not always necessary. For clinically significant isolates, CLSI recommends broth microdilution or Kirby-Bauer disc diffusion assay for those species listed there. So some clinically significant ones. Pseudomonas accounts for the largest percentage of all non-fermenters isolated from clinical specimens. They're called pseudomonads. They're gram-negative bacilli or coccobacillus. They metabolize in strictly aerobic conditions, motile with a polar flagellum or polar tuft or flagella. They're oxidase and catalase positive, are usually oxidizers of carbohydrates, and usually grow on McConkey agar. They're found in moist environments, pools, hot tubs, catheters, and humidifiers in hospitals. So Pseudomonas aeruginosa is the #1 cause of what's referred to as hot tub rash. So sometimes bacteria can overgrow in a hot tub, and this particular type of bacteria can survive at pretty hot temperatures. So 42 degrees Celsius plus, it can grow actively in those temperatures. If the bacteria overgrows, the bacteria can then get on the skin, and it causes this wicked rash all over the surface of the body. Catheters are another main issue. So if we have any indwelling catheters, any respiratory devices, they can form these membranes around them, and they are encapsulated. So they have a capsule that will help protect them. And that capsule not only helps hide them from your immune system, but it helps prevent the access of antimicrobials to them. So it makes them a bit more antibiotic resistant and a bit more of an issue. Reservoirs include plants, soil, tap water. They're uncommon part of the normal bacterial microbiota. Causes mild illnesses in healthy people and severe infections in those with weak immune systems. It accounts for 5 to 15% of all healthcare acquired infections, especially pneumonia and bacteremia. This is the leading cause of healthcare acquired respiratory tract infections. So any type of ventilator or respiratory aid can get contaminated with this, and it can cause some significant respiratory infections. We find this most commonly in patients that have been hospitalized because they're in the hospital for a reason. There's something going on with them. They're sick, and that sickness or that trauma is making them immunocompromised. And that immunocompromised state kind of predisposes them to getting more infections, especially with Pseudomonas aeruginosa. Other clinical infections include wound infections, pulmonary, nosocomial urinary tract infections. Nosocomial would be healthcare acquired. Endocarditis, bone infections, ear infections, eye infections. Poor prognosis and colonization factors. So poor prognosis factors, it can lead to septic shock, granulocytopenia, inappropriate antimicrobial therapy, and the presence of septic metastatic lesions. Patients mechanically ventilated may quickly become colonized. So this is what it looks like on gram stain. And some virulence factors, as I mentioned, the capsule is significant. Here's what it looks like on a plate. A lot of times you'll see this like greenish color if you incubate it on a regular like TSA plate or a nutrient agar type plate. Members of the Pseudomonas fluorescent group include P. aeruginosa and others. These members produce a yellow-green or yellow-brown pigment called bioverdin. Most strains of P. aeruginosa produce the blue water-soluble pigment called pyocyanin. Pyocyanin plus bioverdin equals the blue-green characteristic color. Sometimes on qualifying exam questions, you're going to read these questions about which of the following organisms produces a pyocyanin or bioverdin type color that's distinctive on media. So that's why this one's highlighted for P. aeruginosa. Other times I've seen it referred to as a grape-like odor. Which species has a grape-like odor? Or they'll describe a tech is working up a gram-negative organism. When doing the whiff test or the waft test, they will smell a grape-like odor. What organism is the most likely cause? Grape-like odor is associated with Pseudomonas aeruginosa. There's some various characteristics that are listed here. It is innately resistant to many antimicrobial agents. It's typically susceptible to aminoglycosides, semisynthetic penicillins, and third and fourth generation cephalosporins. Multi-drug resistant infections can be fatal in critically ill patients. So, Pseudomonas aeruginosa is a big deal. It's really difficult to treat once you get it, and it's difficult to recover from those types of infections. So it is a clinically significant pathogen in healthcare environments. Moving on to Acinetobacter. It's now a member of the family Moraxellaceae. So we already talked about Moraxella back when we talked about Neisseria, but only two species commonly seen in clinical specimens, A. baumannii and A. loflii. Epidemiology, ubiquitous in the environment, associated with hospital equipment, particularly ventilators, humidifiers, and catheters. They cause opportunistic infections, account for 1 to 3% of all hospital-acquired infections, second most common isolate to P. aeruginosa. P. pseudomonas. Infections are common in patients with weakened immune systems and coexisting diseases, urinary respiratory tracts, and peritoneal fluids. A list of diseases that are associated with Acinetobacter species would be UTIs, pneumonia, tracheobronchitis, pneumonia, endocarditis, septicemia, meningitis, cellulitis. Gram stain morphology. These are gram-negative coccobacilli. Sometimes they're seen as cocci. Acinetobacters can resist decolorization and retain crystal violet, leading to misidentification. Gram-negative cocci and stains made from blood cultures. It grows on McConkey agar and it produces this purplish hue. A. baumannii. Isolates are often resistant to many antimicrobials, variable susceptibility to aminoglycosides and beta-lactam plus beta-lactamase inhibitor combinations, often resistant to carbapenems. A. loflii is generally susceptible to most all antimicrobials. Centrophomonas maltophilia. It's the third most common non-fermentive gram-negative bacillus isolated in clinical laboratory. It's ubiquitous in the environment, water, sewage, and plant materials, very common in hospital environment, blood drawing equipment, disinfectants, transducers, and other equipment. Initially regarded as a saprophyte or colonizer, not part of the normal human microbiota, respiratory involvement common in hospitalized patients, pneumonia, endocarditis, wound infections, and bacteremia, rarely meningitis and UTIs. Identifying characteristics and treatment, so colonies may appear bluish on McConkey agar, oxidase negative, non-fermentative gram-negative bacillus, positive for catalase, DNase, esculin, gelatin hydrolysis, and lysine decarboxylase. An E-test or agar dilution can be performed for antibiotic susceptibility. Treatment, it's usually susceptible to SXT drug of choice. Burkholderia cepacia. At least 18 distinct genomic species. There are nosocomial pathogens, nosocomial again found in healthcare. Pneumonia, particularly in CF and chronic granulomatous disease. CF is cystic fibrosis. Endocarditis, UTIs, osteomyelitis, dermatitis, wound infections, hospital environment, fluids, aesthetics, nebulizers, detergents, disinfectants. It grows on most media. It may lose viability on SBA in 3 to 4 days without transfers. Selective media available to reduce growth of other gram-negative bacilli. Non-wrinkled in appearance, may be helpful to distinguish it from P. stewartsi that also produces a yellow pigment, can produce a non-fluorescent yellow-green pigment and may diffuse into the media. Often produces a weak, slow positive oxidase reaction. Most strains oxidize glucose. Many oxidize maltose, lactose, mannitol, lysine decarboxylase, and ONPG positive, ornithine decarboxylase and nitrate to nitrite negative, motile by means of polar tufts of flagella. Intrinsically resistant to aminoglycosides and polymyxins. Many strains are resistant to several beta-lactam antibiotics. Isolates may be susceptible to chloramphenicol and others. Variable susceptibility to carbapenems. Resistance can develop rapidly while a patient is being treated. Burkholderia malayi. It's rare in humans, but it is a respiratory zoonotic infection primarily affecting livestock, horses, mules, donkeys, considered by government agencies to be a potential bioterrorism agent. It causes Glander's disease. Only one case in the past 50 years that was caused by a laboratory accident. Endemic in parts of Africa, Asia, Middle East, Central and South Americas. Because of the virulence, susceptibility tests should only be performed by approved laboratories. So you have to send this test out to a reference laboratory typically. Recommended drugs to consider using are listed below. Pseudomalayi causes melioidosis. Symptoms vary and are nonspecific and may include pneumonia, abscess formation, and or blood infections. Diseases can initially be mistaken for other diseases such as tuberculosis, and proper treatment may be delayed. Non-fermentative wrinkled colony exhibit an earthy odor. Ash down medium. Work on suspected cases should only be done under a biological safety hood. So this is kind of what they look like, very unique appearance. Gram stain, bipolar staining, you would identify using MALDI or PCR. Treatment, although isolates may be susceptible in vitro to many antimicrobial agents, the clinical response to therapy is usually slow and relapses are common. SXT and fluoroquinolones seem to work well clinically. And you can review the points to remember